Welcome to this Winemakers Research Exchange virtual sensory session. My name is Joy Ting. I'm the research enologist for the Winemakers Research Exchange, and I am joined today by Vitor Guimarães of Morais Vineyards and Jordan Kennedy, who's representing Wine and Beer Supply. Today, we're going to be talking about the various aspects of Brettanomyces infection in wine, and we'll be tasting an experiment using reverse osmosis and selective absorption or adsorption to remove some of those metabolites. Before we kind of jump into all of the content for the day today, though, I am curious to hear from you all. So have you ever experienced quality reduction in one of your wines due to Brettanomyces infection? So I think Jen is going to launch a poll for us here so that you can kind of weigh in on that. I will promise you that this is anonymous, so I won't be calling on anybody to explain to talk further about it. But have you ever experienced quality reduction in one of your wines? I think one more. We've got just a couple more responses here. Might have one more participant or so. Okay, so Jenna, you can go ahead and end that poll. It looks like you all are very much this may be a well-selected group in the sense that you came here because you've seen this in your own wines, but it is a very common thing. At this point, it looks like we got about 81%. Sorry, this keeps jumping. 81% of you on the call responded that yes, you have seen some quality reduction in your wines. And I would just say that you're not alone. According to the Brettanomyces Economics Survey Project that was conducted through the UC Davis Robert Manavi Institute Center for Wine Economics, about 73% of the folks that responded to that survey said that they indeed had also seen quality reduction due to Brettanomyces in their wines. Interestingly, in that survey, about 9% of people reported a quality improvement in their wines with Brettanomyces infection. And that gets to sort of one of these questions of, is Brett always bad in the wine? It turns out there are a couple of sensory studies out there on that topic, basically where they go and they take a clean wine and they spike it with different amounts of Brettanomyces metabolites. Most of the time, well, there's mixed results in those studies. There are some examples where a small amount of Brettanomyces might increase complexity. There is, however, kind of the biggest thread or the biggest consistency is with a lot of Brettanomyces infection. It's almost always that those wines are scored as having lower quality along the way. So one other poll I would like to do, and Jenny, you can go ahead and launch the second poll. True or false? You may have heard this along the way. Somebody might have told you this at some point. That there are some good strains of Brett that don't cause any spoilage. So true or false? There are some good strains of Brett that don't cause any spoilage. And then along with that, we'll also ask this other question: how is it that you can tell if you have a Brettanomyces infection? So this would be a good time to sort of list some descriptors that might be associated with Brettanomyces infection. But also, if you have other ways that you can detect or that you yourselves are determining if you have a Brettanomyces infection, feel free to put those in there as well. Okay, we have about half of you responded so far, so we'll get you a little bit more time. I realize this free response question takes a little bit longer to type into. Just 30 more seconds there if somebody is just finishing up typing. Okay, Jenna, why don't you go ahead and end the poll for us and then maybe share the results so that we can see, because we'll need to kind of see the end of that free response question.
Are you able to see the free response answers?
I'm not.
Okay.
Folks on the call, are you able to see the free response answers? If somebody could, so Jordan, can you nod yes or no for us so that we can see? You're
I
able
can
to,
I
okay.
can see mine. I just see
Okay.
mine.
I can only see mine, Joy.
Okay, Jenna, can you see them?
I can see them, yeah. We have
Okay,
a lot of responses. Do you want me to
maybe,
read
yeah, if you could
them
read them
off?
off or summarize if there's ones that we have a lot of them.
Sure. It looks like I'm seeing Band-Aid a lot. Aroma more than anything, barnyard, diaper, musky, horse stall, clove, smoky, leather, menthol, medicinal, horse's sweat.
Okay, yeah,
Mhm. Through scorpion analysis,
okay, yep, so
4EP,
that's one of those
4EP
detections.
tests,
Yep.
earthiness, lack of fresh fruit character,
Mm.
dull masking of the wine character, like wet paper over a bar graph,
Okay.
rubber.
And rubber. Okay, great. Thanks, Jenna. So you guys really got a great list going on there, right? You've hit a lot of the highlights. There's all these sort of aroma compounds that come out that kind of tell us this smells like Brett along the way. And then somebody did also put in some of those quantitative analyses that you might use, especially if you suspect you have a Brett infection, and that would be 4EP, 4EG analysis or scorpions. And we'll touch on those in a little bit. So if we kind of want to take a step back, I'm going to kind of address both of these questions a little bit together also. So, you guys hit on a lot of these common descriptors that we end up with: Brettanomyces, Band-Aid, medicine, horse, barnyards, smoky, spicy, sweaty, cheesy. Mousy also is one that can be associated with Brettanomyces. Several years ago, when they were developing the Brettanomyces aroma wheel, which you may have seen coming out of UC Davis, they basically took like 99 different strains of Brett that they had in their freezers, and they grew them up, and they kind of then did this survey of what are the aroma active compounds that Brettanomyces can produce. And it turns out there's a whole bunch of them. They identified 44 of them, and I realized this is pretty small type. You don't need to see all of them. It's really just to show there's lots of different metabolites that Brettanomyces might produce. And some of them they're not all bad. So for example, the one circled in orange here, ethyl octanoate, gives us some really nice fruity descriptors like pineapples and apricots. Not all of the Brett strains produced all of these. So some of them produced just a couple, some of them produced most of this. However, there were a couple of compounds that every single Brett strain that they tested did produce, and that includes the 4-EP, the 4-ethylphenol, and the 4-ethyl guaiacol. Those sort of become our standard bearers for Brettanomyces infection, because basically all of these Brett strains are producing 4-EP and 4-EG. 4-EP is the compound that's most likely to give us like that Band-Aid medicinal quality. 4-EG is the one that's more likely to give us like cloves and spicy kind of characters. Now, when you kind of look at if we get back to the Brettanomyces aroma wheel, eventually they sort of put together all of these different descriptors that were associated with Brett into this aroma wheel to help us sort that out. So when we think about the relationship between 4-EP and 4-EG and all of these different descriptors for Brett, we can remember that it's not just the 4-EP and 4-EG that are causing that, but a number of those other compounds put together that get us to like horsey or to barnyard or to yogurt or to some of these other compounds. But if we kind of zoom in on that 4-EP and the 4-EG, it kind of makes sense that those are the ones that are almost always, or that are always there, at least in that particular survey. These are things that basically are metabolites that are produced from precursors that are found in almost in all of our grape varieties. So they come from hydroxycinnamic acids, and the 4-EP sort of comes from Guimarães acid. The 4-EG comes from ferulic acid. But these are phenolics that are found in the pulp and in the skins of our grapes. So it turns out even white wines, if they get infected with Brettanomyces, they also have the potential to produce these compounds. The Brettanomyces brings to the party, they bring sort of these two enzymes that are going to convert those hydroxycinnamic acids in kind of a two-step process into our Brettanomyces metabolites. So basically all of our grapes have this, and all the Brett strains have this. So they all have the potential to produce these compounds. The one thing that is different sort of between in different Brett infections would be the ratio of 4-EP to 4-EG. So for example, Pinot Noir has about a 3-to-1 ratio of 4-EP to 4-EG, whereas Syrah has about a 23-to-1 ratio of 4-EP to 4-EG. And that is part of the reason that sometimes our Brett infections have a little bit different presentation in different varieties. So for Pinot Noir has a relatively higher amount of 4-EG. So it has a little bit more of that spicy or clovey kind of component to it, whereas Syrah has a lot less of that 4-EG. So it's a little bit more straight up Band-Aid or medicinal. So the other thing is, so I guess getting back to the question there, is there a good strain of Brett that doesn't cause spoilage? Well, if you consider 4-EP and 4-EG to be spoilage, all of the strains of Brett are really going to produce that. But again, there's the component of how much of that versus those other aroma compounds that they're producing might still give us differences in how that's expressing in different infections. So the other thing to think about is that our Brettanomyces infections aren't only about 4-EP and 4-EG, and they're not entirely just about those aroma compounds. There's other sensory impacts of having Brettanomyces infect your wine. One of the primary ones is an increase in acetic acid. And this really has to do with the metabolism, this sort of direction of metabolism at the end of glycolysis. So at the end of glycolysis in Saccharomyces, acetaldehyde is converted to ethanol most of the time and to acetic acid once in a while. Unfortunately, Brettanomyces, that sort of trigger point there, shunts it over to the acetic acid portion of the program more often. So sort of glucose for glucose, there's going to be about 4 times as much acetic acid produced by Brettanomyces than produced by Saccharomyces. Now, most of the time we don't have that much glycolysis going on by the time Brett gets there, but once it does get there, it's much more likely to give us more acetic acid. We also have a reduction in fruit character really by 2 different mechanisms. The first is that Brettanomyces produces enzymes that cleave fruity esters. So those aroma compounds that make our wines seem fruity, the Brettanomyces is going to come along and just sort of start chopping those up. So we have fewer of those compounds. But the other thing has to do with our perception of fruitiness. Studies have shown that our perception of fruitiness declines if you spike that wine with sub-threshold amounts of acetic acid, 4-EP, and 4-EG. So all of those compounds basically make your brain less likely to register those things as being fruity. It really has to do with kind of your brain's ability to suss out the signal versus the noise. And then the last thing that we'll highlight today is a loss of color. So Brettanomyces, in addition to bringing esterase enzymes, it also brings in enzymes that cleave monoglycosylated anthocyanins. And what that basically means is that it does that cleavage and it leaves your anthocyanins more susceptible to bleaching or to taking on a colorless form. So in general, there's about a 30% color loss with a Brettanomyces infection. I bring this all up because as we talk about remediation strategies that are going to remove 4-EP and 4-EG, we need to remember that there's these other things that have happened to the wine as a result of Brettanomyces infection that maybe aren't necessarily remediated with these strategies. So in that same UC Davis survey that I cited before, 93% of the survey responders said that they took some kind of active measures to control Brettanomyces. So I'm going to kind of put that back out to you all. Most of you said that you'd seen Brett at one time or another. So I'm going to go ahead and assume that you're doing some things to try to prevent Brettanomyces infections in your cellar. So I'm going to ask you to go ahead and maybe write into the chat, what strategies do you employ to prevent Brettanomyces? What are some of the things that you're doing in your cellar that maybe you would do differently if there were no Brettanomyces, or at least things that you think are going to help you to be able to remediate Brettanomyces, or I'm sorry, prevent Brettanomyces? And while you guys are writing into the chat, I'm going to figure out how to see my chat. There we go. So I anticipate there's going to be lots of different answers here. So go ahead and just write in whatever you're doing there, and we'll take a look at it in just a minute together. Yep. Okay. These are very interesting. Thank you all so much for sharing these. We have just another bit here while they continue to come in. Excellent. You guys hit a lot of the good points here. So I see lots of folks talking about sanitation. And of course, sanitation is like the first line of defense against any of these things. I'm going to go ahead and go to my slide here. I see folks, Bola says quick malolactic fermentation. So a lot of the most common time that we're going to see Brettanomyces infection is really kind of during malolactic, when the competition gets lower and lower. It's harder and harder for other organisms to find anything to eat in there, but our Brettanomyces comes in there. So quick mallows lead to a faster SO2 at the end, so we can hopefully deal with it a little bit there. Low pH and adequate SO2, those all go together. A lot of folks mentioning, especially with sanitation, the importance of using steam when you're dealing with your barrels. I think we've all sort of seen those horrifying papers that talk about the fact that Brett can sort of get up into the barrel staves and live up there. And so in order to really clean those barrels, we have to have our cleaning method also get up into the pores of the barrel staves. The common number right now is steaming at least 12 minutes. Of really hot water for a period of time also is helpful along the way. I also see chitosan there. So chitosan is an antimicrobial. Essentially, it's a fining agent. So there are definitely ways to be using that as a preventative measure. So some folks are putting that in their post-mallow and like kind of leaving it in there. And it does seem to have some prevention there. We'll see later on, it's also kind of useful as a curative effect as well. Let's see. Timely regime of SO2 use. And I think SO2 itself is I have a few more specific things to say about SO2, but SO2 is really an important tool that we have to manage those populations of Brettanomyces. So I kind of put up this list here, and it's really beyond the scope of our time today to go into the specifics of each of these. But I found that for myself as a winemaker, as I was reading through these papers, I wanted to find some really practical stuff. So I did kind of put together a document that I'll put on the WRE website that talks a little bit more about each one of these prevention measures. Things like how long you need to be steaming your barrels or how hot things need to get in terms of cleaning, some of those kinds of things. So that will be on the WRE website in the next day or two to give us a little bit more a little bit more detail about that. But I think what I do want to do is just talk a little bit about the fact that, there's lots of different things on our list here because there's no one silver bullet to deal with Brettanomyces. Brettanomyces is generally very tolerant to the conditions that we find in wine. It does a really good job with high alcohol, so it's not inhibited by 15% alcohol. It still seems to be able to divide and metabolize. It's very acid tolerant. So one study said that in 94% of the strains that they tested, that Brettanomyces was tolerant to pHs as low as 2. So that doesn't mean that it's in, it's its favorite place to be, but it does mean that it can grow and survive along the way. So pH doesn't really seem to affect it in that way. It doesn't require many nutrients at all. It turns out it's a pretty bad competitor. So we see it in very, very, very low amounts in grapes because it just can't really compete. But as fermentation goes on and there's less and less there, it's really good at kind of hanging around and then scavenging what's left over. That includes very, very low levels of sugar. So even at 275 milligrams per liter, so 0.28 grams per liter of sugar, it can produce 1,000 milligrams per liter of 4-EP, which is way above our sensory thresholds. It's also fructophilic, and it can metabolize other unfermentable sugars for Saccharomyces. So basically, the paper that I was reading said there's always enough sugar out there to feed the Brettanomyces population, no matter how dry you get your wine. It can survive on and reproduce on very low levels of nitrogen. So one paper cited 6 milligrams per liter of yan, which is almost undetectable, was enough to support a population of 10 to the 7 cells per milliliter. Just for reference, that's about an order of magnitude higher than what you're normally inoculating your barrels with for regular fermentation. So that's a lot of cells. Part of that might be that it can use nitrogen sources that Saccharomyces can't use and therefore are left over in the wine. And then getting to SO2. SO2, it is relatively SO2 tolerant. Most of the recommendations that we see out there recommend that you use between 0.6 and 0.8 milligrams per liter of molecular SO2. So remembering that molecular is the antimicrobial form. Just to put that in context, if your wine has a pH of 3.6, your free sulfur would need to be between 38 and 50 to hit that 0.6 to 0.8. Most of our wines, especially our white wine or our red wines, have higher pH than that, and that means we would need more free sulfur. So it's really hard to get to that number of 0.6 to 0.8. There is evidence that at about 0.4 milligrams per liter, you start to sort of move these cells into a little bit different state that's called a viable, not culturable state. Unfortunately, there is also some evidence that they continue to produce metabolites even when they're viable, not culturable. So you might be able to slow them down a little bit, but you probably aren't stopping them from being able to produce these things. But it does slow them down, and it is helpful to maintain your SO2. The other thing there is if your SO2 gets depleted over time, as it does when we age in the barrels, eventually these cells can emerge from that viable, non-culturable state and start to really reproduce in larger amounts. And then the most shocking thing that I've heard recently, I was listening to a webinar from somebody who works with the Australian Wine Research Institute. They did a survey of the Brettanomyces populations they had sort of in their freezers from 2000 to 2018. And what he said is that when they grew those up and they started looking at their SO2 tolerances, they saw SO2 tolerances as high as 1 milligram per liter of molecular sulfur, which means that there's just no way that our sulfur can really necessarily control all of these Brett populations. There might be a strain out there that can get through. The other thing they found is that when they looked at the really old ones in the freezer versus the newer ones that they were finding in wineries, that they were finding that they were getting more and more tolerant along the way. And that makes sense. We're sort of putting in some selective pressure so that the more and more tolerant ones survive, whereas we're sort of keeping the other ones from being able to compete as much. So SO2 is very helpful with regard to managing Brettanomyces, but don't think that it's the only thing that just maintaining your SO2 is going to be able to keep Brett at bay. So with that, I think that the take-home message is there, you know, obviously there's just multiple different hurdles that we want to put up along the way with all of these things that you guys have listed in terms of sanitation and managing our microbial populations through inoculations and prompt malolactic, using our sulfur wisely, racking when we're supposed to be racking, those sorts of things in order to try to keep those populations as low as possible. So the question is, you know, even when we do all of these things, sometimes Brett still happens, right? Again, 80% of you said you've had it, you've seen it already, and probably it's out there just lurking, waiting for us. So what do we do if we do detect Brett in one of our wines? So when we think about treatment options for Brettanomyces, there's really kind of two things we need to keep in mind. We need to think about removing the Brettanomyces itself, but then there's also removing the metabolites. So even if you're able to remove the Brettanomyces, those metabolites are stay behind. Those are sort of two separate processes. For removing the microbes, one of the tools that we have to do that is chitosan. So I think Jeremy mentioned using chitosan as a preventative measure, which is definitely there. You can also utilize chitosan at a little bit higher dose in order, and what it does is it will sort of help the Brettanomyces cells to bind and settle into the lees. And then with those curative rates of chitosan, you're told to sort of rack off. So with prevention, a lot of times people are not racking off. If you have an active infection, use that higher rate, wait your 7 to 10 days or whatever the package tells you, and go ahead and do that racking so that you can remove as many of those microbes as possible. You get a several orders of magnitude decrease in your Brettanomyces population when you treat it in that way. The other thing that you can do is you can filter, right? We think about filtering before bottling. Filtration will remove those cells, whether you're doing that right before bottling or if you're doing that after a racking before you go down for long-term aging. If you know you have a Brettanomyces population, filtration will help you as well. But then what about those metabolites? If you've had your Brett active, you've been able to smell it, you know it's there, that 4EP, 4EG, and all those other guys, all those other metabolites are in there. And filtration doesn't get rid of them, and chitosan doesn't get rid of them. Well, one thing that I came across as I was doing some reading for this was that there are a couple papers talking about using yeast lees as a fining agent for Brettanomyces metabolites. So it turns out that, you know, the cell walls of yeast are very absorptive of a lot of different things. And there are a couple papers out there using yeast lees to sort of put into a Brett-infected wine and then looking at the before and after of what was happening with those metabolites. And what they basically found was that they could get out about 25 to 30% of the Brettanomyces metabolites by using yeast lees. It looks like there's a particular manoprotein in the cell walls of the yeast that are binding to the 4EP and 4EG and taking those out of the wine. So if you have to do a little bit of remediation, that might be a gentler way of kind of dealing with it. But if you have a lot of Brettanomyces, you're probably going to be looking at doing some reverse osmosis with some selective adsorption in order to remove larger concentrations of Brettanomyces metabolites. So because this is the method that we're going to be looking at today is doing sort of this RO with selective absorption, we'll go ahead and zoom in on that picture just a little bit. Now, this particular picture actually shows you what would happen if you wanted to remove acetic acid, but it's really the same principle along the way. So if we start with our wine in our tank here, it runs through this high pressure pump, goes into a chamber that then basically through a process of initially osmosis, but then some pressure to get it to go even further against its concentration gradient. It's going to sort of do this permeation. And what's happening here is that the membrane itself has very tight junctions on it, so only very small molecules get through. So the larger molecules like your tannins and your polysaccharides and hopefully most of your aroma compounds don't ever go through this membrane, and they just get recirculated back up into your tank. However, the small molecules do come through, and that includes water, includes alcohol, includes acetic acid, but it turns out 4EP and 4EG are small enough to get through that membrane. So whatever goes through here, that's called permeate. And that permeate then gets shunted over to this sort of selective column. And again, this picture is showing what would happen if you had acetic acid to remove. So in that case, we would use an ion exchange column. It would bind up the acetic acid and hopefully let everything else flow through to join back up with whatever got retained and go back into the tank. Now, in the case of 4EP, 4EG, we don't want to use that anion exchange column because that's not going to bind our 4EP and 4EG. Instead, we need to use a different medium that's going to bind specifically to 4EP and 4EG. And I shouldn't have said specifically, because basically what it's doing is it's binding to any of our sort of hydrophobic things that are going across that column. In that case, it does bind 4EP and 4AG. But the question with any of these techniques is what else is it binding along the way? So there was a study that was done very early on with this method. The study came out in 2005, and they basically looked at the chemistry of the permeate before it went through that column and the chemistry of the permeate when it came back out on the other side. You can see there are all kinds of different things that get bound along the way. For sure, the 4EP and the 4EG get bound, and we can see we have up to a 75% reduction in our 4EP and 4EG after several turns through that particular column. But you can see there's a whole bunch of other stuff on this list as well. Some of them you probably wouldn't mind getting rid of. Some of these acids that have aromatics of rancid and rancid butter, those can go ahead and stick and not come back into our wine. Our C6 alcohols, those are the things that give us the green character that we call tomato leaf kind of characters. Go ahead, those can just stay on that column and not come back as well. But you'll notice there's this whole category of esters on the top. These are a lot of our fruity compounds as well. So when we're sort of doing these things and we're worried about stripping the wine of aromatics, it's these kinds of compounds that we want to keep an eye on along the way. So, so that sort of brings us to our experiment for the day today. And so today this experiment kind of came about about a year and a half ago. And to sort of explain what happened here, I'm going to go ahead and bring in Jordan and Vitor. So if you guys can unmute yourselves. This experiment was done, the one that you tasted was done on Merlot from Morais Vineyard. And then we also have data on another lot of wine from the Tannat of Morais. So Vitor, tell us a little bit about your Merlot and your Tannat for this experiment. What vintage were they?
Sure.
You know, how long were they aging? How long into the process did you know that you were going to need to do some remediation?
They're both 2021s. Just a disclosure, I got a little bit of a cold going on, so not only the accent is going to be on the way, my nose is not working today, so I'm going to sound a little nasally. So they were both 2021 wines. The Merlot is estate grown. The Tannat was fruit purchased from a different vineyard, a different winery. Actually, in 2021, for some reason, I had 4 different wines with Brett issues. It never happened to me. I think one of the reasons was some barrels that I probably bought that were used that I shouldn't have, but learning the hard way. So I would say about, I was trying to pull up the info, but I would say about a year from, I would say somewhere in 2022, maybe summer 2022 before harvest, I started noticing something wrong with the wines. I'm kind of sensitive to Brett. So, you know, when I was pulling samples every now and then, I could just open the barrel and you know, I would get a whiff of the 4EP, 4EG. And I think around that time, I reached out to Jordan because we had worked earlier to remove some VA from a wine I had. And you know, Jordan told me that they had those new the carbon filters that they just acquired recently, and they wanted to try it. I remember I talked to you, Joy, in person, I believe. Was it during the Eastern Winery or was it during Virginia Vineyards Association meeting? I remember we discussed
we
something.
talked in person, but those 2 meetings are so close together. I don't remember when it was, but yeah.
Yeah, and actually I remember that you said we should probably do something together, and because you were interested in doing some more research about Brett and 4AG, 4EP. And then I reached out to Jordan. Yeah, sorry.
Go ahead. No, go ahead.
Oh yeah, and then I reached out to Jordan, and and Jordan, of course, always with his busy schedule, found some time for us. And he came by first time. We fixed the wines. I feel like the other three wines were immediately fixed that day, and the Merlot also seemed like it was, but a few weeks or maybe like a month later, I was pulling samples again, and at this point the wine was in a tank. I didn't put it back in barrels again because I was getting it ready for bottling. And I just was pulling a sample, just tasting, and I felt that something was not right. And then once again, I reached out to Jordan, and he came by second date to work on the Merlot again.
Yeah. Yeah, so I mean, I think the timing of this, you and I spoke at that meeting, and I think Jordan and I had heard that you had started to do some of this, and we did want to kind of look at it and just say, what are some things that we need to think about if we're going to utilize this service? How do we make sure that we make the most of having a visit from Wine and Beer Supply in order to get this done? And so thank you so much, Vitor, for letting us kind of come alongside as you were doing this and being willing to share this with us, the experiment itself. So and so Jordan, tell us a little bit about kind of how things worked with the treatment at Morais. So tell us a little bit about the different steps that you had to do along the way.
Okay, so the crossflow, the pre-filtration is really important because if you have a wine with high turbidity, you're going to clog your RO membranes really, really easily. So you always need to have a clean wine. So sterile filtered, you want to get the bacteria out of there anyway. And then the RO process is recirculating within the same tank process. And as you split that permeate stream away and you treat it, it's important to make sure that your treatment, that permeate is actually getting cleaned up before you put it back in the tank. But you only have a small percentage that passes that membrane as you run. So it's kind of a continuous recirculation with a smaller percentage treatment going on. And you monitor along the way and decide when you think it's good enough anyway.
Yeah, and we'll talk about a couple of strategies for that too. So one question for you, Jordan, a lot of times we'll see this today, we'll sort of get this like how much was treated versus the volume of the tank. So that amount that's treated is what's actually, you're able to measure what actually goes over that membrane and becomes permeate, right? So you're measuring the volume of permeate is so it's not that you only kind of treated 20% of the volume of the tank. It's that once you went all the way through, 20% of the volume went over that permeate membrane.
Correct.
Okay, great. That helps me understand it much better. Thank you. So when Vitor and Jordan were doing this experiment, basically we took samples at a couple of different times. So it looks like my sample number is not showing up here. Sorry. Oh, sorry. So let me see. So we did have a sample, and I thought I had a tag here, but it looks like it's disappeared in the ether. But we did take a sample right after crossflow, and then before crossflow in this tank. There you go, you can see it. Sample, and that would be we're going to call that T0. So before we even started. And then we had another sample after all of it had been treated the first time around, and we'll call that sample T1. And eventually we'll also have a sample T2, which will explain why we have a sample T2 in just a couple of minutes. So just to take a look at kind of the chemistry along the way. Yep, there we go. If we look at the Merlot, we did take samples for wine chemistry as well. And we can see that there's just a little bit of a little bit of movement in the wine chemistry itself. Now, none of these are measures that we would expect to change at all with this treatment. And probably the difference that we're seeing here is that, again, Vitor had a couple of different wines that were being treated, and there is kind of a reservoir of wine inside of that, inside of the machine itself. And so some of these small changes probably just had to do with mixing with the reservoir of wine that was already in that machine. So we had one sample of Merlot that in this case, it looks like we had about 20% of it that was treated in this round. And then there was also, as we said, a tank of Tannat that got treated. That was a much smaller tank. And in that case, about 120 liters out of the 700 liters were treated. And we see the same thing with the general chemistry. Again, a little bit of movement here and there, but nothing huge here. And again, we wouldn't expect to see big differences in this along the way. One of the other things when we do a lot of filtrations is thinking about what's happening with the oxygen permeation. We did have a DO meter on site there. So Vitor and or Jordan took these DO numbers. And what we can see is that crossflow itself, it does introduce some oxygen along the way. And we talked about this a little bit with our, when we did our session on closures. That the dissolved oxygen that was introduced during crossflow had really been consumed by the wine by the time the RO was done about 2 days later. And this change in about, it looks like about 0.5 milligrams per liter of dissolved oxygen. That's not a huge change, but it is something if you have a really, really sensitive wine. The RO itself does not seem to be introducing even as much as crossflow. It introduces just a little bit. But again, all of the different things that you normally would do in a filtration to manage the oxygen are happening here as well. The Tannat, we had a little bit higher increase in the dissolved oxygen with crossflow. And talking to Jordan and Vitor, it sounded like maybe a little bit of air got in the line there. So that might have been more about, dealing with the really small little variable tank rather than the crossflow itself. And again, being Tannat, that dissolved oxygen was consumed by the wine very quickly and pretty consistently by the time we got there. The other set of samples that we took along the way was a Brettanomyces panel. And ETS, this is just a panel that you can order from ETS. And there's a couple of different pieces to this panel. And so I did want to talk about what each one of these panels or pieces is telling us. So the first one is scorpions, and somebody mentioned in the chat that scorpions are one of the things you can do to tell if you have a Brett infection. Scorpions are a molecular technique, and so basically these are going to detect any of the cells that are out there, whether they're in that viable, not culturable state, or if they're in an active state. So the scorpions themselves are the primers that they use for that molecular technique are species specific. So they kind of run these 3 different kinds to see if you have these different types of yeast in the sample that you send them. That Brettanomyces panel also does include this wine yeast culture portion. And first of all, you do see there's something there of presumptive Brettanomyces. So this is where they're actually growing them up on culture plates. So the presumptive Brettanomyces portion here will tell you how many of you have, how much of your Brett is not in that viable, not culturable state, which we think is probably more active in producing metabolites. But the other thing that the ETS website says is it kind of tells you how effective your SO2 regime is. So if you have a pretty high number in scorpions, but you have a low number in your culture, that means you've at least shunted a fair number of them to that viable, not culturable state. But again, as we said, they still are there and they might be doing some activity, and they certainly can do some activity once the SO2 gets a little bit released. But then there's all these other yeast in there, and you might be wondering, why do I even care about those on a Brettanomyces panel? And ETS basically says we want to look at these other things to see how heavy is your microbial load in general. Because if you're going to go ahead and age this wine and you have all those microbes in there, eventually those are going to autolyze. They're going to put a bunch of nutrients into the environment. And so it's good to know if you have sort of this big nutrient influx that's getting ready to give up if you're aging on your yeast lees, or if you have a relatively clean wine. So they kind of give you the rest of that information along the way. And then, of course, we have our Brettanomyces metabolites, our 4EP and our 4EG. And I've gone ahead and put a general threshold number here for both of those. For 4EP, general threshold is usually about 600 micrograms per liter, whereas 4EG is about 200 micrograms per liter. I would say with these thresholds, that's very dependent on the wine that you're in, but those are some general numbers to keep in mind. So if we look at the Merlot before it was treated, we can see that there was a little bit of molecular signature of having some organisms in there, but it was pretty low. So whatever Brettanomyces infection had happened there probably had already kind of been racked off as Vitor came off of the barrels and went into the tank and did some other things. not a whole lot of signature there, and certainly nothing that was coming out in terms of a culturable effect. But we can see he really does have quite a large signature here of 4EP and 4EG. So certainly somebody was active there for a while. If we look at after treatment in that first round, we see basically nothing in terms of our microbiology except for just a few cells of Saccharomyces. And that's just a nice reminder that, you know, the wine is only as microbiologically clean as the vessel we put it into. Our wineries are full of Saccharomyces all over all the time, and no matter how well you clean them, it's hard to get rid of all of them. So this just shows us there's still a little bit of Saccharomyces hanging around. There's nothing to be worried about in that wine with regard to Saccharomyces, but there still is just a little bit of a signature of that. But the other thing we can see is that in just about, I think we said it was about a 20% run of the wine here. We have about a little bit over 40% reduction in the 4EP and the 4EG. Now, Vitor and Jordan, you guys were sort of basically like smelling this wine as you went along to determine sort of when to finish. So tell us a little bit about kind of the sensory when you started and why you decided to stop when you decided to stop.
Yeah, well, like you said, we were tasting as we went along. Jordan told me he didn't have a lot of experience with Brett, so we kind of like tried to get kind of a sense of, you know, we talked a little bit about it, you know, like what descriptors we came up with, both of us, and what we decided was that every 5 to 10% Jordan would give me a shout, and we would, you know, pull a sample of the tank out of the tank and taste it again, both of us, and we would you know, discuss a little bit between the both of us, you know, how the wine was tasting and if it was getting better. We had the sample that we pulled previously from before the treatment, just to, you know, to compare, and we just, you know, that's basically our procedure, and we decided to stop, or I decided to stop when I felt that the wine was tasting pretty clean. And in hindsight, it's very scary because I did not realize how quickly you get desensitized to the blind to Brett. And that's why Jordan had come back the second time, because at that day, and it was a long day also, I feel like at a certain point we were both a little tired. You know, because we were pulling samples, you know, every 10 to 15 minutes and tasting them, and we were, you know, with all the equipment running and everything. And it's kind of scary that at a certain point both of us were not tasting anything weird with or wrong with the wine, and then a few days later, you know, I tasted the wine again, and it was still there, you know.
And I think this is, yeah, we talked about that. Like, this is one of those sort of really hyper practical things that go along with having a new technique to kind of work with is, you know, we don't have an in-house lab test to determine how much Brettanomyces is left in there, right? So if you're trying to, you know, change, you know, if you're doing acid additions and you want to know if you've really hit your pH mark, you have a pH meter, you can measure that. But I mean, basically we got these numbers back about the same time that you went back a couple of weeks later. It took a couple of weeks to even get these numbers from ETS. So you can't really use that as a quick turnaround. So I would say that the Tanat basically followed the same pattern here. We once again get a really nice, you know, we get rid of all of the microbes along the way. We have a fair amount of reduction in 4EP and 4EG. But again, at the end, we're still kind of above, at least above threshold for those things. So as Vitor Vitor and I were talking about this, and Vitor was like, I think I'm going to have Jordan come back. I happened to be talking to Emily from Veritas, and she made the suggestion that, you know, sometimes with some of these metabolites, Brettanomyces is one of them, and I think some of our reductive metabolites are those as well, that it's really helpful to have a super clean wine to use as reference. Sort of like having those, you know, coffee beans when you're doing a tasting to just sort of cleanse your nose a little bit and then go back. So So Jordan and Vitor and I talked about that, and we'll see in just a minute when we look at the round two. That was sort of added into the protocol when they did round two. But again, that's one of the reasons that we do this as an experiment and sort of follow things as we go along, is to say, are there some super practical things that might be really helpful in having success with this on the other side of it. So before we see what happened in round two, though, I did want to show you one other set of chemistry here. And that was really what's happening in the permeate itself. So the next set of things I'll show you, we basically took a sample of the permeate before it went through the column and after it went through the column. And the whole idea there is to say, how good is that column itself at taking out the 4EP and the 4EG? So if we look at that, and I think, and Jordan took these samples at two different time points along the way. One was early on in the run, and one of them was later on in the run. So that kind of tells us, is that column getting saturated over time? So at time number one, the permeate before it got treated had about 3,500 micrograms per liter of 4EP and about 573 micrograms per liter of 4EG. We look at what happens after treatment, there's almost nothing left. We get a huge reduction in both of those. So at least early on, the column is doing a really great job of taking those out. That second time point, which was later on through the run, and we can see we've had a reduction in the 4EP and the 4EG going in, because again, it's that recirculating tank. Afterwards, once again, there's almost nothing left. So we again have a really very effective removal of those metabolites as they go over that column. So we can see it's really being effective in doing that. But again, because we sort of started with these at the end of round one, there were a fair number of metabolites left over. Vitor decided to do a second round of treatment, at least on the Merlot. And so we said, let's go ahead and go along for this ride as well and just see what that chemistry looks like as we go. So the one thing, there's sort of two differences here in round two. The first is that reference wine that we talked about. And the second one is that this time around, they ran more of it. So the first time it was about 20% of the volume. Second time it was about 60% of the volume. Is that right, guys? About 60%?
Hey, Joy, could I ask a question?
Of course.
On your time one and time two of your first round, is that minutes? Is that hours? Is that many hours?
That's a good question. Jordan, you were the one that took the samples here. I think it was just early in the run and later in the run. But I guess the question is how long was it taking to do that run of, each of those tanks of Merlot that were at like 2,400 liters each?
On the first visit, we didn't really run any single tank more than about an hour on the larger Merlot vessels. So the flow rate is controllable. We could have done more volume of treatment faster, but we kind of settled in on by taste. For me at the beginning, when you first start to run, you want to taste that permeate coming out of the carbon. And when I kind of found that, guarantee, like, yes, this permeate is improved, it's better, then we went with that. But I think about an hour.
And what was the size of the lot? Just trying to get reference points.
Those, Vitor, do you remember the exact volume?
The Merlot, the total was, I had two tanks at 2,300 and 2,400 23 and 2,400 liters.
And I think for the experiment, that first round, the samples all came from one of the Merlot tanks.
Yeah, I think we
didn't
picked one of
follow
the tanks,
other
yeah.
Merlot tank.
Yeah.
So the total volume for time one and time two on this tank would have been 2,400.
Yeah, we picked one of the tanks. Yeah.
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 Does that answer your question? Good. Okay. Yeah. All right. So the second time around, and that was a good segue, because the second time around we did end up Vitor and Jordan just treated that Merlot lot, but we followed both tanks the second time around just to get kind of a replicate as we went around. Everything else was same in the process though. So if we look at what happened with the 4EP here, I've gone ahead and given you the 4EP that we had in the first round. And again, we had about that 42% reduction with about 20% of the volume going through. The second round, the first tank, you can see it starts right about where we kind of left off on the first one. So that was the tank that was being run the first time. And with 60% of the volume, we saw about a 37% decrease in the 4EP. That second tank started out with a little bit more 4EP to begin with, but again, we have about a 48% reduction. Now, we kind of might wonder, why do we have essentially kind of less bang for your buck in terms of time as you go along? But we have to remember that this is a concentration-based process. So if we go back to that paper from 2005, they had this nice graph, I think, that shows it, that kind of the longer you run, the less 4EP or 4EG you're dealing with, kind of the less proportion you can take out over time. Because again, you just sort of have less hitting those charcoal particles, so you can kind of take out less proportion each time. So that process that we see happening is what we would expect. Unfortunately, it does make it a little bit hard to decide like how much is being taken out over time, because instead of a linear reduction, you kind of have this exponential curve that you're dealing with. If we look at what happened with the 4EG, it's kind of the same thing. We still had, we once again had a little bit kind of less proportion for the amount of time that was spent, but we still do have a fair amount of reduction happening over time. The other thing to notice here is that even at the end here, we still, these wines are still kind of above threshold for both of these metabolites. And again, realizing that like that's not, that you don't have to necessarily get below threshold. The idea just is a kind of where we are relative to that threshold. We once again did run those permeate samples at the beginning and the end. And in this case, we have the beginning and the end of that first tank. And then we did also run permeate just at the end of the second tank, because that was about the same as the end. We didn't need to do a beginning of tank 2, because that would be the same as the end of tank 1. So in this case, what we can also see is that, you know, of the permeate going through that particular, through that column at the beginning of the run, we once again have that really super efficient reduction in the 4EP, and you'll see in the 4EG along the way. But as that run continued to go, we had kind of less efficient removal over time. And again, that might be that we're just starting to saturate some of those sites on the charcoal itself. You know, and that makes sense. There's only so many binding sites in there. And so eventually you might start to saturate some of those sites. So unfortunately, what that leaves us with is that it still is pretty difficult to be able to kind of predict exactly how long you're going to need to run that column in order to remove a certain proportion if you know your metabolite number to begin with. Okay, so that's the data that I have to show you. There's one more bit of data that I want to show you, and that's what you have in the samples that you have in front of you. But before I actually tell you which ones are which ones, I do want you guys sort of, if you have your wines in front of you, I'd like you to just put them in, maybe make a guess of the order here. So if you can sort of take the wines you have and see, if I tell you that the 3 wines that you have in front of you, one of them was taken before that first round, before crossflow. So that is like our beginning number. The second one was taken sort of at the end of round 1. And then the third one was taken at the end of round 2. So you're essentially putting these in order of the amount of Brettanomyces metabolite that you can detect in them. So see if you can guess which one is T0, which one is T1, and which one is T2. Okay, I'll give you just another few seconds to do that, and then I'll tell you which ones are which. Okay, So I'll go ahead and show you. You'll have to remember which group you were in. You might have to look that up, or it might be, hopefully it's on your piece of paper. So I'll give you a minute to look at this before I make it go away, and that way we can, but in order to kind of talk about our sensory impact impressions, I am going to ask you, if you have your camera off, to go ahead and put your camera back on. After I give you just another moment looking at the flight here, I'm going to take the slides down so we can see each other as we talk about our sensory impressions. Okay, everybody good? I'm going to go ahead and stop the share. All right. There we go. Hello, everybody. So yeah, again, if you could put your cameras on, if you're in a situation where you can put your cameras on, that's always better for discussion. And let me find my, here we go. Okay, hello, everybody. Thank you for joining us today. Okay, so were you able, so when you think about kind of that order of what we would predict in terms of the Brettanomyces metabolites, who was able to put them in the correct order? Maybe just raise your hand and we can see who put them in the correct order. Okay. All right, wow, great. So it did seem to make a big difference in terms of what was in terms of sort of those kind of big chunks between or big differences between them. How did the wines, what allowed you to put those wines in order? Was it just the Brettanomyces metabolites, or were there other things going on there that you were like, this sort of seemed to be going on? So somebody tell me about your impressions in putting them in order. Either, again, if you want to just unmute yourself, this is a good time for us to just talk that way. But if you're not in a place you can do that, you can feel free to go into the chat. So what were the things that you were picking up as different as you went through that progression?
Joy.
Mhm.
I lucked out because in mine, I started with the lowest amount and then went to the highest amount. So I was able to kind of see a progression of, fruit decreasing and Brettanomyces, like, the Band-Aid and a lot of the chemical quality just get worse and worse in each glass. So for me, it was just the increasing impact of Brettanomyces in my glasses.
So was it mostly the Band-Aid, or did you notice some differences in fruit too?
Both, yeah. As the Band-Aid increased, as that intensity increased, I lost anything from the treated wine.
Okay. Other impressions? There are other things that you would observe about what that was smelling or tasting like along the way?
From me specifically, were you asking me?
Or no, anybody
Okay,
else.
good.
Sorry. Yeah, no, Emily, thank you for piping in there.
What I found interesting was the reduction of intensity in the treated wine and more spice character. But
So the reduction of Brett intensity or
no,
overall
about
intensity?
aromatic intensity as a whole, and so you have less intensity, but you have more refined elegance of aromatics.
Yeah, I think that's a good point. Did anybody else notice a reduction in the overall aromatic intensity, not just necessarily the Brett metabolites, but overall aromatic intensity? Yeah.
I just, Joy, if you don't mind, just want to add, following what Luca said, that was one of the reasons we stopped treatment so soon on the first time on the Merlot, was because it was the first time doing this for both of us, and I was really afraid that since it was a carbon filter, that we were going to remove a lot of the fruit from the wine, and I actually felt during the treatment that it was getting less intense, the fruit character, and so I was afraid of stripping too much of everything. And on one end, you're stripping the 4AP, 4AG that you want to do, but then at the same time you're removing, you know, the good things about the wine, you know, the aromatic intensity and all of that.
Yeah. And I think that's a big concern that people have about these kinds of methods. Are you going to be stripping those things out? So I'm sorry, AJ, were you about to say something?
Yeah, it's interesting to me because I tasted mine in the same order as Emily for the most part and got the same perceptions. But I really so I was in group 1, so it was 540, and I did this yesterday because I had time. And let them sit with a little watch glass over top of them prior to that, and really got a lot of initial fruit intensity on for what 540, the final product. And noticed that on 119, which was the second one, just very little fruit intensity. But as I let them sit longer, it kind of swapped.
Okay. Yeah. Interesting. And I think that we have that complexity of what's going on with the fruit, right? Is that the 4EP and 4EG is making us, you know, perceptively making that go lower. But again, if we're taking out fruity esters and sort of going through the column, we maybe take them out that way as well. So it might be one of those that, like, yeah, you leave a little bit of metabolite so that you don't process as long because you find some sort of good middle part there that you're happy with along the way. Okay. Any other impressions? Any other ideas in terms of kind of the beginning and the middle and the end? How different were the two treated ones for you guys? Did you find those to be, was there sort of more of a big difference between not treated and either one of the treated, or the two treated ones really seemed different?
Joy, we have a message in the chat from Shai.
Okay.
He was in group 2, which would be, let's see, 569 was T0, had the highest perceptive VA, 4EP, and fruit intensity.
Okay. Okay. Yeah. So they are like kind of all of them were expressing at the same time. So that too might get back to, like, you know, it is more, we do get more complexity when we have more of those things around. It's just a matter of if that's a complexity that we want. Okay. How about the perception of other, like, of VA in general? Did that seem to shift at all, or did that sort of stay the same for the whole lot?
I noted it more into the sample 327 because in the sample untreated with more complexity is masks. The second one, the 327, I noticed a little bit more, and I noticed it less on the 871.
Okay. Yeah. Any other thoughts about the overall volatile acidity? Again, remembering the column doesn't take that out for us, but as Luca said, like, sometimes those other things impact how much we're perceiving that.
I have a question for Luca.
Okay.
Luca, in regards to your aromatic intensity,
Yeah.
you know, I agree with you. We lost aromatic intensity. Do you think the aromatic intensity is, you know, the quote unquote good part of Brett, where it brings so much aromatics to it? And would you rather have aromatic intensity, maybe not that high? Would you rather have that, or would you rather kind of have that quote unquote stripped wine? It's not stripped, but, you know, a decreased aromatic intensity.
I think that a way in somewhere in between it will be my preference. Okay, so I could even think of treating if I have a tank, I treat 80% and retain some in the back. I can add on, so therefore I don't run all of it through, and I keep something that is more integral with all its elements, and then you can tweak it, add on, versus if you stop too late, you cannot go back, because I think some of these compounds, they're trapped in the treatment. So, but something in between. I prefer the T1 for my flavor than the T2. It's more pure, but it has less character and is leaner and a bit more is driven more on spice than fruit for me. And I like it better in the middle, I would say.
I'd be interested. I'd love to hear from those of you what your preferences would be as well. So maybe I'll ask you to raise your hands. I'll give you a T0, a T1, or a T2. So who likes the untreated one the best? Anybody? Okay. Okay. All right. Anybody like T1? Who else likes T1 the best? We know Luca does, but anybody else like T1 the best? Okay. All right. And then T2, you have to, and then you can't not vote. People are not voting. So anybody that, all right. Okay. So, and I think that gets at that idea that like there's different balance points in there, and depending on kind of where you are with things. Okay. I'm seeing a few more coming in here. So I've got a few more T1s along the way. Kendall says T2. All right. Okay. Other reasons. So for those of you that prefer T2, what is it about T2 that you're like, this is for me, why it's the best one? My T2 people, why did you like that one the best? Cleaner? Okay, Melanie says because it's cleaner. Yeah.
I feel like if we're gonna go through all of the 4 EP removal, I think, you know, for me, the least one was more preferable.
Okay. Yeah. Who would have gone further? Okay. Okay. Yeah. And that's the hard part, right? Is figuring out when to stop, right? How far to go, when to stop. And even as Lucas said, do you hold some back to put it back in if you feel like you've stripped too much? So, okay.
Yeah, just if it serves as a warning, I mean, like I said before, I got to a point where Brett was not something that I knew what it was, because I just became blind to it, nose blind to it. And it was kind of weird. So I feel like if somebody ever has to do this, get a group of people to help you out. good wine tasters, the people that are capable of, that know Brett and that are able to be very objective, as objective as you can be, because I feel like this is very subjective, of course. I feel like some people are going to be more sensible to Brett than others. But yeah, you get somebody that's familiar with Brett and can, you taste one time, the other person tastes the other time, and kind of take turns in tasting.
Yeah, multiple noses is always that's always a good strategy on things for sure. So any other for those of, oh, go ahead, Luca.
Yeah, well, the hard part is are we tasting as winemakers or are we tasting as consumers? That's the big difference. I try never to be searching for a little defect. The VA was a little high. I mean, I have to think of outside of that when I make choices. And that's kind of the way I think. Other people think it's very different. They want a perfect wine. They want to have everything. But sometimes you don't get that, and try to correct a wine that has perhaps a characteristic trying to go completely away. It may work in our choice idea, and but I'm not sure if for the consumer it works.
That's a good point. Is that sometimes we're looking for faults when the consumer is looking for something delicious. An earlier comment, Shai brought up that both of the treated lots had kind of this nice Dr. Pepper effect to them, that sort of added to the character as well. So it's important to realize we do get some of that along the way also. So just a couple of things kind of as take homes, and I know that we're getting toward the end of our time here, but we did sort of see that we can do what we want, whatever we can to prevent Brettanomyces infection. It's a lot easier to deal with a wine that's cleaner, and we can make those decisions. We also, in order to get this treated, we do need to remove both the cells and the metabolites. But there's also some of those other impacts on the wine that the Brett sort of leaves its signature behind. Regardless of all of our great efforts to try to keep this from happening in our cellars, it still does happen, and it happens to all of us, really because this is a microbe that has very broad tolerance to a lot of the conditions that exist in our cellars. And so pretty much Brettanomyces is one of the community of microbes that we find in the cellar. We also saw that in terms of that remediation using RO and absorption, it's helpful to have that clean wine as a reference. But then also, as Vitor said, that it's really helpful to have multiple noses, or maybe just even bring that wine to like somebody in the tasting room, that when you're tasting room employees, and say, hey, smell this for me, to sort of try to determine when is a good time to go ahead and stop doing that as well. We didn't really get into the cost of this as much, but that's another thing to consider is, you know, whether the differences that you're smelling and you're tasting are worth the cost of having the service come out. The overall cost of the treatment includes both the crossflow filtration and the RO and adsorption treatment. That treatment is about $1.70 per gallon without the crossflow, and then you have the crossflow treatment on top of it. So you can kind of decide if it's making that a big enough difference in terms of what the quality potential of the wine along the way. But before we do sign off today, I did want to make sure also to make sure that we thank Jenna for all the work that she's been doing. Jenna has been with us for 2 years now as our exchange coordinator, and she is leaving us at this point to go take another job outside of the wine industry. So I just want to say a very heartfelt thank you to Jenna. Many of you have had the opportunity to get to know her a little bit and work with her in this last couple of years. She has done a fantastic job. So much of her work is kind of behind the scenes, so you don't really get to see all of the important things that she's been doing, but she has been instrumental in putting together just a number of systems to help things to run more smoothly here at the Research Exchange, to help keep us up to date a lot better than I could ever do on my own. And she's just brought her wonderful giftedness to us in that way. So Jenna, thank you so much for all of the work that you have been doing for us. We will miss you, but we do wish you the best as you kind of move on to this next chapter of your life.
Thank you. Thank you so much. And it's been such a pleasure working with all of you. The winemaking community is such a down-to-earth, great group of people, so I've really enjoyed it.
Well, we know you'll stay connected because, of course, your wonderful husband, Jeremy, is also in part of our community. So we know we'll continue to see you at least, you know, around at events and that sort of thing. So
I hope so.
glad
Thank you,
for that.
Joy.
And I would also want to say that, you know, we will be rehiring Jenna's position. I will probably be sending out a job description through the WRE email list in the next couple of hours or maybe tomorrow morning. But if you do know of somebody that is very well organized, that is interested in learning about the various production aspects of wine, but also getting sort of a front row seat to all of the experiments that we do along the way, we would love to have you encourage that person to apply, and there will be details of how to do that in the email that I send out. All that information is already available on our website as well. So if you want to check that out, we are really we're really looking for a suitable replacement, but also somebody to come in and bring their set of ideas and talents to this part of the Research Exchange as well. So we do appreciate your help in that if you do know of somebody that would be a good fit. So with that, let me go ahead and thank you all so much for coming today. We are so grateful to the Virginia Wine Board for their funding of our project. We are so grateful to Vitor and Jordan for sharing this experiment with us, for taking all of these samples and all of this data along the way. And I'm thankful to each one of you as well for providing your sensory statistics, for participating in discussion today, and just for sticking around and helping us to think through these various aspects of things. If you do want to do some additional experiments on Brettanomyces, please let me know. And we can always kind of start planning already for the next round of things. So thanks so much for that. We'll stick around a little bit. If you have individual questions to ask, you can feel free to do that as well. So thanks, everybody.